Welcome to Evolutionary Biology. Today, we are going to explore one of the most fascinating phenomena in nature, speciation. Have you ever wondered how the incredible variety of life on Earth came to be? How did we get from a single ancestral life form to millions of unique species, from microscopic bacteria to massive blue whales? The answer lies in the birth of new species. In this lecture, we will build our understanding of this process from the ground up. We will learn what it means, how it happens, and even how to measure it mathematically. Let us dive in. To understand how new species form, let us start with a simple analogy. Imagine a large group of friends who all speak the exact same language and interact every single day. Now, suppose a massive earthquake suddenly splits their city in half, creating an impassable divide. They can no longer visit each other. Over hundreds of years, these two separated groups will naturally invent new slang, alter their pronunciation, and eventually speak completely different languages. If they ever met again, they would not be able to understand each other at all. In nature, this is exactly how new species are born, from a single ancestral group. Building on our language analogy, let us look at what happens to living organisms. When a single population of animals or plants gets physically separated and stops mixing, they slowly begin to change. Over long periods, these changes accumulate until the two groups can no longer mate with one another. This magical, slow-motion process of splitting one species into two distinct groups is what we formally call speciation. In evolutionary biology, speciation is defined as the evolutionary process by which populations evolve to become distinct, reproductively isolated species. It is the fundamental mechanism that generates the tree of life. Now that we know what speciation is, we must ask, what exactly is a species? According to the biological species concept, a species is a group of actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. This means that if two individuals can come together in nature and produce healthy, fertile offspring, they belong to the same species. If they cannot, they belong to different species. Therefore, the entire process of speciation hinges on interrupting the flow of genes between populations, which sets the stage for them to drift apart genetically. Since speciation requires interrupting gene flow, let us explore how populations actually diverge once they are separated. Without the sharing of genes, genetic divergence begins to accumulate through three main drivers: mutation, genetic drift, and natural selection. Each isolated group adapts to its own unique environment. Once these genetic differences become so significant that individuals from the two populations can no longer produce viable, fertile offspring together, speciation is considered complete. This beautiful process bridges the gap between microevolution, which involves small genetic changes within a population, and macroevolution, which is the grand scale of life's diversity over millions of years. Understanding this bridge between micro and macroevolution is absolutely crucial because it explains the incredible biodiversity we see on Earth today. Without speciation, our planet would only have a single, uniform type of life form, rather than millions of beautifully adapted species. In real life, scientists use the principles of speciation to protect endangered species and manage wildlife reserves. By studying how populations diverge, conservationists can figure out which groups of animals need to be protected together to maintain healthy gene pools. It also helps us understand how new agricultural pests emerge and allows us to trace our own human origins. Knowing why speciation matters leads us to a practical challenge for scientists. How do we know when speciation is actually occurring? We can't just look at their physical appearance because sometimes completely different species look identical to the naked eye. Instead, scientists need a way to measure how genetically different two populations have become over time. To do this, we use a mathematical tool called Wright's Fixation Index. This index compares the genetic variation within a single group to the total genetic variation across all groups combined, allowing us to quantify exactly how far along the path of speciation two populations are. This mathematical tool we just discussed is expressed through a specific equation. The formula for Wright's Fixation Index is FST equals HT minus HS, all divided by HT. Let us break down what these symbols mean. FST represents the fixation index itself, which measures the genetic divergence between subpopulations. The variable HT stands for the expected heterozygosity, or genetic variation, in the total combined population. The variable HS represents the average expected heterozygosity within the individual separated subpopulations. Conceptually, this formula calculates how much of the total genetic variation is due to differences between the groups rather than within them. If FST is close to 0, populations are freely mixing. As it approaches 1, they are completely isolated. Now that we have our formula, let us see this with a concrete example. Imagine a population of frogs recently split into two groups by a newly formed river. Biologists find that the total expected heterozygosity for all frogs combined is 0.50. The average expected heterozygosity within the two separate groups is 0.40. We need to calculate the fixation index to determine how much they have diverged. We start by substituting our values into the numerator, subtracting 0.40 from 0.50, which gives us 0.10. Finally, we divide this difference by the total variation of 0.50. Our final answer is 0.20. This moderate value tells us the populations are on their way to becoming different species, but speciation is not yet complete. Let us look at another scenario to see what completed speciation looks like mathematically. A species of bird is blown by a storm to a remote island, separating them from the mainland flock for 1,000,000 years. Researchers find the total genetic variation across both groups is 0.60. However, the average variation within the groups is only 0.06. We want to evaluate if these island birds have become a completely new species. We plug our values into the formula, subtracting 0.06 from 0.60 to get a numerator of 0.54. Dividing this by the total variation of 0.60 gives a final fixation index of 0.90. This extremely high value means gene flow has completely stopped, and the island birds have almost certainly undergone complete speciation. Having calculated the genetic divergence, let us now visualize the physical process that causes it. On screen, you are looking at a diagram illustrating allopatric speciation, which is speciation driven by geographic isolation. At the top, we see the initial state, a single, uniform original population of green beetles freely mixing in their environment. Moving down, a physical barrier forms, in this case a river, splitting the beetles into isolated population 1 and isolated population 2. As time passes, we reach the bottom stage, divergence and evolution. The barrier has caused reproductive isolation. The population on the left remains green, adapted to the original environment, becoming species A. The population on the right has turned brown, adapted to a drier environment, becoming species B. We have covered a lot of ground today, so let us review the most important ideas. First, speciation is the beautiful, continuous process that generates the tree of life by splitting one species into two or more distinct species. Second, this process fundamentally relies on stopping the flow of genes between populations, which allows them to adapt to different environments or drift apart genetically. Finally, we learned that scientists can measure this divergence using mathematical tools, like the fixation index, marking the critical boundary where microevolutionary changes build up to create entirely new branches of biodiversity. You now have a solid understanding of how Earth became filled with such a magnificent variety of living things.